We're going to go through all of the procedures and setup for the digestion lab. Notice today I'm wearing gloves. It's going to be really important when you're demonstrating and when your students are up here that they wear gloves this week, because we're using a lot of pH buffers that are in extreme pH ranges. So they won't initially burn the skin, but if they are left on the skin, they can be very caustic. So we need to remind the students, and I want you guys to demonstrate wearing gloves. All right, it's very important. Obviously, there's a lot of experiments to do in here. All right, so half of the battle of teaching this lab is getting people organized and keeping people on task. Okay, so if you notice in your TA guide, I have some things you need to do when you first come in. So let's go through those first. For the preparation of the lab, when you come in, remember you're supposed to be here 30 minutes early. First thing you need to do is turn on all of your water baths, especially the hot water bath, because it'll take about 30 to 45 minutes to get up to temperature. Okay, the other thing you'll need to do is down here on this end, we're going to have a cold bath. So if you take this downstairs with the test tube rack inside, leave the thermometer, fill this up to the top with ice from the first floor ice maker, and then you'll bring it back and you'll take a 600 ml beaker, fill it with ice, and put ice into the water for the cool bath or the cold bath. You'll also take some water and fill this to where the top of the water and the ice bath is up to the top of the test tube rack, and the test tube rack is submerged. Okay. Once that's done, make sure that you have a thermometer within it, because the students will need to record the temperatures of that bath. Okay? Make sure that all of the water baths are turned on. So this one, normally we don't turn this one on, but for this experiment, we are using this one at 37 degrees. So you'll flip the switch up on the front. And these, especially the hot ones, check the water level, because you can see as it's steaming, we're going to lose a lot of water volume. So make sure that before you turn these on that the water level is up above the test tube rack, and check it periodically through lab. If you notice that it's starting to get low, don't add water in the middle of lab, because it's going to drop the temperature and it'll mess up the experiment. Okay, but make sure you do it at the beginning of lab and check it at the end of lab. There's several of these enzymes that need to be kept in the refrigerator, as well as the heavy cream, which is our substrate. We don't want that to rot, so make sure that you go and get all the solutions out of the refrigerator, and they're all listed in your TA guide. Okay, and that's in the common closet. The other thing is the spectrophotometer. Up here, we're actually going to have two spectrophotometers. Right now, I have this one plugged in for the purpose of this meeting, but we'll need to find a different spot for this, probably at the other end of this station. Okay, find the spectrophotometers and turn them on, and leave them on for at least 15 minutes before lab. Okay? Okay, so that's what you do when you first come in. Once the class starts, before you even start your quiz, we need to assign jobs to the students. Okay, so the first thing you're going to do is you'll need one person that's going to be giving the saliva sample. Okay, and they're going to be spitting in a funnel for 10 or 15 minutes. And so it's better to start this before the quiz and give them a piece of gum, so during the quiz they can be collecting their saliva sample. Okay, so you'll want to demonstrate how to do this. So we have these gauze squares, and all of the extra gauze squares are up in that front cabinet. Okay, so at the beginning of lab, you need to make sure each station has one. But they're going to take their gauze square, and you're going to open it. If you leave it four-fold, the spit won't go through it. The main reason why you need this is to get any food particles or any of the mucusy stuff out of the spit. We want the liquid. All right, so they're going to take this and make sure you have a glass funnel, not a plastic one, because this is a physiological or a biological waste product with the spit. And you're going to take your gauze and stick it down into the bottom of the test tube, into the bottom of the funnel, so you have a nice collection place for the saliva. Then they're going to set it in this Erlenmeyer flask and take a piece of gum. They'll be chewing it, collect the spit, and spit into the tube. Now, they're supposed to get 10 mils, okay, or up to 20 mils. We need a minimum of 10, okay, and then they'll fill out the volume with water. So if they get 15 mils, then you just add 5 mils of water, okay, but we want a total spit volume of 20 mils, okay? So you can imagine it's going to take them a little bit of time to get that sample up. So you want to start that before the quiz, so you're not waiting 30 minutes into lab for them collecting their sample. So once they're done with their quiz and you get going, you'll need to demonstrate and kind of talk about where all the stuff that they're going to need for their lab. So we basically are going to have 4 jobs at the table. A lot of these experiments have multiple test tubes that have similar volumes. Okay, so what we'd like you to do is take one person to the table. Their job is to mark the test tubes. And so if you look in the lab manual at the first page, it says several of the exercises. You have exercise 1A here. It says 2 tubes with lids. And then there's 12 tubes with lids. You can have one person that's going to come up to these containers, take those test tubes out, and measure them with those volumes. Okay, then they'll hand them off to whoever's doing those exercises. It will make things run much more smoothly. Okay, then you're going to assign exercise 1 to one person, exercise 2, and exercise 3 to different people at the table. So all 4 people should have a job. Okay, if you don't have 4 people at a table, if you have a table that has a person that's absent or dropped, then they can divide up the marking duties for the test tubes. Okay, so just then assign each person their exercise and they'll mark their own tubes. Okay, are there any questions about that? Okay, so once the person that's done marking the solutions is done marking, they just need to kind of be the gopher for everybody else. So whoever's lagging behind and setting stuff up to get in the incubator, it's their job to help out. Okay, remind the students though that just because they've been assigned a particular exercise doesn't mean they don't need to know what's going on in the other ones. It's their job to, when in the downtime between measurements, when things are incubating, to discuss what they're doing with their lab members and make sure that they're aware of what's going on in all the exercises, because they're responsible for all the data. The other thing about this lab is they're going to have a lot of test tubes. So if you see a bunch of people that are sitting back not doing much and they have dirty test tubes at their table, encourage them to clean up as they go. Okay, it will facilitate things much better, and you won't have a clog up at the end at the 3 sinks when everybody's washing. For each of the exercises, I just want to run through. If you notice here in your TA guide, you've got lecture topics. Now I listed these here, but we don't really want to lecture a lot because this class takes a lot of time getting set up, and there's a lot of incubation time. So these are there for you to understand the background, and if you have time and you have downtime once things get going, you can stop the class and kind of talk about some of these things, okay? But the experiments, these are important. So I'd like you to take notes along here as I go through these different segments, okay? So let's start with exercise 1. So exercise 1 is a very simple experiment. They're going to be mixing water with oil, and then they're going to be looking at the effects of bile salts, okay? So Danielle very nicely pre-mixed these for us. So they're going to take two test tubes. One of them is going to have bile salt solution. So this is water with bile salts in it and oil. And they'll put a couple of drops of food coloring in. For the control, it's just going to be oil and water. Okay, so when they shake them up, okay, let's look at the control first. When you shake up the control, you guys know oil and water doesn't mix, right? Almost immediately, the oil and the water starts to spread out again and make 2 distinct layers. So remember, food coloring is water soluble. So the water is going to be on the bottom, oil is on top. But the nice thing about the color is it distinguishes between the 2 substances. So you can see on the side, if you've got a droplet there and it's bright blue, that's going to be water versus oil. The oil droplets are going to be clear. But you saw how quickly that separated out. Okay, and if you look at the sides, and I'm going to pass this around in a second, if you look at the sides, there are some droplets here. I'm trying to show you this close up. Yep. There's some droplets here that are water, and then there are some droplets here that are clear. So you have distinct droplets between the 2 liquids in the tube. And that's what you would expect, right? Bile is created in the liver and secreted by the gallbladder, right? After you eat, the gallbladder constricts and it's released where? In the duodenum, right? Okay. And what's special about bile salts? They're amphipathic. Okay, so they have a polar and a nonpolar end. Okay, so what the bile salts are going to do is they're going to emulsify the fat. So if I shake this up and I look, this is important. I want you guys to all see this. Okay, so I'm going to pass this around. I'm going to demonstrate this and then pass it around, and I want you to observe what the students would be observing. So if you look here on the sides of the test tube here, where the droplets, you actually have little tiny droplets of oil within the water. They're not big droplets, they're uniform. They're little tiny droplets. And you also get this layer that looks like shaving cream. Okay, and that's because you've got little bubbles of oil within the water, okay? So pass these around and look at those and observe. So the students are going to observe initially right as they shake it up, but then we're going to ask them to leave it and observe later. So these guys have been sitting here for a period of time, okay? And let's look. The control has two very distinct layers, okay? And they're completely separated. We still have a little bit of water and a little bit of oil stuck up on the side, but within the contents themselves, they separated into two distinct forms. But look at this one with the bile salts. We have three very distinct layers. Okay, so at the top we have the oil, just like we did in the other test tube, and at the bottom we have the water, just like they had, but what's in the middle? What does that look like to you, Hillary? You can take it. Tell me.
The center?
Yeah.
The shaving cream?
Say louder.
The shaving cream?
Okay, it looks like shaving cream. You see the distinct droplet. Okay, so what's happened there? Anybody? Don't be shy. Say louder. Okay, so she said it's been broken down into smaller fat droplets. That's correct, but I want you to be careful with your terminology, because when you say broken down, I know what you mean. But broken down also implies digestion, and we want to distinguish the fact that bile salts are not actually digesting the fat. All they're doing is emulsifying the fat, okay? So they have the nonpolar end that can bind to the nonpolar fat, and the polar end that can interact with the water. And as a result of that, you get these little tiny droplets, and you get a water interface around the fat droplets. Okay, so why would it be important to emulsify the fat?
It gives more surface area for digestion to occur.
Exactly. It gives more surface area for digestion to occur. So remember, the enzymes that we're talking about are water soluble. So the only place that they can get in touch with is where the fat is integrated with the water, is touching with the water. Okay, so increasing the surface area allows for the lipase to get in touch with the fat in the fat experiment, in the lipase experiment. Okay, so we obviously, when you're doing this, teaching this, you don't want to go through all the explanation in the demo. That's part of the student's discovery. So, but I did want to make sure that you knew what to point out in the observations. So are there any questions about that? Do you guys feel comfortable explaining that? Okay. All right. So let's continue with exercise 1. So over here, down on this end, we have all the stuff that you'll need for the fat digestion, or the lipase. Okay. So you'll have the heavy cream, which is that you get out of the refrigerator. All right. And we've added 4 different temperatures. This is the one that's going to be looking at both the effects of bile salts and temperature on fat digestion. Okay. So let's take a step back, because the students have done their worksheets, right? So what is the most common form of fat that we have in the body? Triglycerides. Okay, so lipase digests triglycerides into what products? Okay, say it louder.
Fatty acids and glycerol.
Okay, so she said fatty acids and glycerol. Technically, it's a monoglyceride. So we have the glyceride backbone with 1 fatty acid and 2 free fatty acids. Okay, that's the way the book describes it. All right, but the free fatty acids is what we're worried about, right? Because if you get digestion of fat by lipase, what's going to happen to your pH? It goes down, right? Because we have more acid in the solution. So as a result, we can measure digestion using a pH indicator. And in this case, our pH indicator is going to be phenol red. Okay, so you can see here, it's a nice pretty red color. Okay, it's this color in basic conditions. It turns yellow in acidic conditions. So we want to start this experiment with the fat being fairly pink to indicate that we're in a basic environment, with the hopes that we'll see a color change with digestion. Okay, so one of the key points to this part is that they have to start with a uniform pH and a uniform color at the beginning, because we're using color as a qualitative indicator, but we're also going to be measuring with pH strips. Okay, so we want at the beginning for the solutions to be the same. So this test tube holds the lipids, or the heavy cream, and 20 drops of phenol red. So you notice that this is kind of a peachy color. See that color? This color would not show a very big pH change because it's peach, right? And it's changing to yellow in acid. So we need to be able to start with a basic solution. So what we're going to do is we're going to add a little bit of sodium hydroxide. So notice that up here I've put small beakers in front of the solutions with droppers. Okay, that's so people don't drop droppers into the bigger containers, which happens. I have this out here. Make sure that you don't overfill the sodium hydroxide though, because if it gets really full and somebody knocks it over, it is a strong base. We don't want anybody to burn themselves. Okay, so we're going to add a couple of drops, and I don't want you guys to add this very fast because we want this to turn pink. But if you add too much, then you're going to go too basic. It's not enough. So notice where the phenol red is, where the sodium hydroxide is, we're getting this pink color. Okay? As I shake it, I want that to be a uniform pink to the color of Pepto-Bismol. You guys know what that is? You know, the stuff you use when you got an upset stomach? Okay? Still want it a little bit pinker than that. All right, and I'm kind of going fast because we're just doing a demo here. The students need to be counting how many drops they're adding to get this color, because that will help them. They're going to have 12 test tubes that they're lining up and trying to make the same color. Okay, so if they figure out how many it takes to take one, then they can start close to that value for the next one. All right, and you're going to get a lot closer to the same pH. I'm just right now trying to get this to where it's the right color to start with. Okay, so see that nice bright Pepto-Bismol pink? That's where they need to start. Okay, and all 12 test tubes before they add the lipase. So this should just have phenol red, the cream, and the sodium hydroxide. They should all be the same color pink. Now, if you get somebody that's really overzealous, okay, and they add a lot of sodium hydroxide, which happens, and you'll get one that's like 10 times darker than the other two, okay? You can use that in the control. Just have them wipe off the number on the side and change it to the control tube, because you're not going to digest that, and they can use that as a color comparison, okay? But what we don't want to do is have that be a test one where they're going to add the enzyme, okay? If they have time, if they're not way behind, you can make them remix. It's your choice. But if it's only one tube, that's an easy fix, okay? Because remember, they're doing this color change before they've added anything but milk and phenol red. Okay? Questions about that? Okay, so Danielle's going to be setting up that experiment in a little bit, and we'll take some pictures of the actual results. I don't have them right now. But what we should see, they're going to be looking and comparing the colors at 2 different times. Okay? So what time did they test it first? Have you guys read through the manual? Are you guys following along with me? 15 minutes. Okay, so they're going to incubate it first. They're going to get all the solutions, one of them of each kind. So you have tubes 1, 2, and 3. 1A, 1B, 1C, 1D. Each of the letters goes in a different temperature. Okay? At 15 minutes, they're going to measure, they're going to record the color and the pH at digestion. Okay? So remember, we've got a trial that has bile salts and one that doesn't. So at 15 minutes, you should see a significant difference in digestion between the one that has bile salts and not. The one with bile salts should turn yellow fairly rapidly to indicate digestion, because we've had the emulsification of fat. Okay? And you should see a change in pH fairly rapidly. That's the one we want to record and graph for the effects of temperature. Okay? Because by the time they get to 30-minute digestion, 30 minutes is a long time to be sitting with an enzyme. But in the digestive tract, in the duodenum, the food doesn't stay there that long. Okay? The food is long past in 30 minutes, and most of the fat's already been absorbed. Okay? So at the end of 30 minutes, you'll see a difference in pH, but there'll be a lot of it will be digested. You won't see a huge difference between the bile salts and non-bile salts. I'm hoping that you will see differences with temperature, and the colder temperatures will have less digestion. But when you compare tubes A and B in the same temperatures, they should be fairly equal at 30 minutes. The last substrate we're going to be measuring is BAPNA, which allows us to use trypsin, right, which is a peptidase, all right, to digest BAPNA. The cool thing about BAPNA is that when it's undigested, it's a clear solution. Okay? So when the trypsin comes in and digests it, it cleaves off a piece that turns yellow. All right? So when you see digestion, it'll go from a clear solution to a yellow solution. All right? So white light is all the wavelengths, right? So how do we perceive yellow then? Yeah, so some wavelengths of light are absorbed and some are reflected. So the colors that we perceive as yellow are reflected and enter the eye, but some are absorbed. Okay? So we can use this to measure the amount of substrate that's in there by using a spectrophotometer. Okay? Because the amount of light that's absorbed in particular wavelengths is going to be directly proportional to the amount of solute that's in there. Okay? So the more digestion we have, the more yellow your solution is going to be visibly, but the higher your absorbance is going to be. Okay? So really quickly, I'm going to show you how to use a spectrophotometer. I'm assuming most of you guys have already used one. This is just a standard spectrophotometer. The on-off switch is on the back, and you should turn this on for 15 minutes before you use it. The other thing that you'll need is some chem wipes or a paper towel. So let me just grab one real quick, because we don't want any fingerprints on the blanks. Okay? So to do this, first thing you want to check is they can change the mode of this by clicking that. So if it's on T, that's for transmittance. We don't want that. We want absorbance. So if you click on the mode, the light should be next to the A. Okay? Now, we're putting through a known frequency of light that's absorbed by the BAPNA byproduct. Okay? That frequency is 430. So you adjust this knob until the line intersects 430. Okay? Now we need to tear it out. So you know what I mean by tear it out? We need to zero it. Okay? So we're going to open this, wipe off around the sample, make sure that there's no debris in the sample or fingerprints on the side. And you're going to slide that in there all the way down until it stops. Then you're going to close it. So to zero it, you're just going to press this button over here, and it's going to blink. It says BLA. It's going to blink that way for about 10 or 15 seconds. Okay? And then it should come up 0.000 when it's zeroed and blanked. Okay, so there it is, 0.000, and it's stable. So at that point, I can remove this blank. And when they do the test tubes, they're going to be doing test tubes 1 through 6, and test tube 6 is a control. It's all going to be distilled water. That's what they're going to use for their blank. Okay? Now, in a normal experiment, they're going to have tubes at 5 different pHs. I just have one here for demonstration purposes. So when they do the sample, again, we want to wipe off the sides, no fingerprints, all right? Set it in here and close it, okay? Now the absorbance is going to read up here. Now notice the number is shifting quite a bit. So we want to get it to where it stabilizes. It may change the last digit a little bit, maybe varying, but when it starts to sit around a constant number, that's when you record that value. Okay? Any questions about that? Okay, so the number here is what they record. So this is stabilizing around 0.732. Okay, then they'll remove the sample. And I would suggest that they tear or they zero between samples. Okay? Is it time to go? Okay. Are there any questions about that? Okay, so common problems. Common problems with the starch experiment. First, I forgot to show you the indicators for this. So let's talk about starch. So we have two different indicators here. One of them is Lugol solution that turns black in the presence of starch. And then this should be Benedict's, which turns kind of a rusty red precipitate in the presence of sugar. Okay? So I have preset this up where I have a drop of sucrose and a drop of starch in this. And I'm going to take a clean dropper and drop some Lugol solution. So we're going to put 2 drops in each of these plates. So if you notice here, the well that had sugar in it has no color change. It's an iodine solution. Lugol's is an iodine solution, so it'll be this brown color. Okay? But in the presence of starch, what color is it?
Olive, kind of black.
Yeah, it's black because it's a precipitate. So the iodine slips in between the pieces of the starch and it falls out as a precipitate, as a black precipitate. Okay? So they're going to use this to compare for a positive starch test. Okay? They'll wash this and they'll use this same plate when they're incubating their saliva sample with the starch. Okay? Now, the most common problems that we have with these is if you notice, Reed set this up a little while ago and it's been incubating, and notice the starch settles out on the bottom and you get kind of this starch pill that's in the bottom. Okay? And it sticks and it gets hard. The longer that it's in the solution, the more dense this little pill gets. So it says in the lab manual for them to use the Vortex. So this is a Vortex. You guys ever used these before? Okay, there's one over here too. All right. So these are set up to be pressure turned on. So if they're turned on, they need to be on touch. When you push down on them, they vibrate and they make a noise. Okay, we have one on each side. Here's one here. This one actually has a adjustment for the speed. You want it at about middle. If you go too high, we might splash out. Okay, so when they get these out, before they take them back to their table, they need to put them in the Vortex Er to break up the pill and suspend the starch. Okay? If they don't do this, they'll have variance in the amount of starch that they pick up with the dropper. Okay? So they need to go back to their table and they need to stick their dropper in at about the same level of the solution. So if they take one from the top, they need to take them all from the top. They don't want to take one from the top and one from the bottom, because even with it gone through that vortex, there's still, you can see, it's clearer up at the top and it's dense, it's thicker down at the bottom. So where they take their solution out is going to be important. What we should see with that experiment is salivary amylase, because amylase comes both in the saliva and in the pancreas, right? But what pH is our mouth? Well, it depends, really. It's a range. We would expect it to be close to neutral, but it can be acidic. If somebody has reflux, they'll have a little bit of acid. Okay? If their mouth is basic, some people have basic mouth. They do. A basic mouth tends to not have very active enzymes, and so don't tell your students this, but if somebody has a really high base, like a 10 base saliva, they're going to have periodontal disease usually, because it facilitates the growth of bacteria and their enzymes don't work as well. We expect the range, the normal range of pH in the mouth to be around between 4 and 7, usually closer to 7. Okay? Now, as a result of that, we would expect to have the most starch digestion in those ranges on your plate. Okay, you should have less precipitate in the 4 or the 7. And you know, I don't know how you feel about this, Heather, but if the students are curious, have them stick a pH paper on their tongue and see what the pH of their mouth is. You know, when they ask, so what should be the optimal pH? And say, well, how do you figure it out? Well, one way to figure it out is to take the pH paper and stick it in their mouth. Okay? I make sure that if you do that, though, you pull out a new piece and the part that they're picking out is clean and hasn't been on the outside of the pH box. Okay? But they can do that. Or spit on something. Stick it in their spit solution. You can have them do a pH paper in their spit solution if they're curious. That way it doesn't actually have to go in their mouth. okay? But we would expect the most digestion to occur at that pH, okay? Now, the starch plates, sometimes you get pretty wanky results depending on how much, if they're uniform and how much starch they're picking up, okay? So we followed up with the Benedict's reagent, okay? The Benedict's reagent reacts with limit dextrins and some sugars. It's fairly, it's not very specific for sugars. It reacts with a lot of them. But when it binds with the carbon ring, it forms that rust-colored precipitate. Okay, so the solution, the Benedict solution by itself, this is Benedict's with starch in it. So you see the white layer at the bottom. That's because the starch is settling out, but there's no color change. Okay. We'll show you a completed result. This one hasn't changed yet, but you'll see precipitate down in the bottom that's kind of a rust color. Now, the top of your solutions will still stay blue most of the time unless there's a lot of sugar in there. So sometimes it's more subtle in that the whole solution will look a little bit more yellow because the precipitate is giving it that color. Okay, but look down at the bottom, the precipitate will be stuck around the bottom of the test tube. And it's a qualitative measure. They're going to be looking at it and making a judgment based on color, which one has the most. So we really want to compare those colors closely. I am going to put together a post-lab workbook and a post-lab movie with the explanations, but I'll let Heather go through the effects of temperature and pH on the shape of an enzyme, because the important part of this is as food moves through the digestive tract, we're going to be encountering different pHs. Okay, so stomach, obviously, what's the pH there? It's about a 2. Okay, and then as the food bolus passes into the duodenum, you get the addition of bile salts and all the pancreatic juice, right? So what happens to your pH? It goes up. It goes to a more basic, between 7 and 10. So in a little space of about 6 to 8 inches of the digestive tract, you have a huge shift in pH. You also have a huge shift in the active enzymes that are there. Okay, so this is a little bit different approach to enzymes than we've done in the past because we're trying to link it back to the digestive system. So the more of that kind of like questioning that you can do with the students, the better.